Okay, so this is a program that can be used. Now you can download the program and use it if you wish, but there's a Streamlit app here that they've linked to now that you can just input matrices into here to try and get the format for the Topaz input. Okay, so you can go and have a read through that and find out what it all means and what it all goes. But essentially what you got here is instructions for the use of Topaz, so it does kind of go through that process with us. Last time round, we discussed how to get a Z matrix from Dash. Do you know how to do that, Sadia? Or
I have used Dash, yeah.
You have used Dash. Okay, so if you've used Dash before, then what happens is that let's just what we're able to do is we're able to try and use Dash, but it basically we can do it, we can use Dash, or we could actually just use it as a way to convert files or convert coordinates into the correct system. So we can go through for each of your powder patterns, go through this process of viewing data and then going through determining peak positions and so forth.
Mm-hmm.
But what we can do, by doing that, you get a .sdi file out, which is kind of all the information that you've got to that point. Otherwise, you can skip to this simulated annealing structure solution.
Mm-hmm.
Okay, so here what we're doing is we're putting in a molecule of our system, and then it inputs it into Dash for Dash to do stuff with. Now, we're not actually trying to solve the crystal structure here necessarily. If we are, we are, and that's great. If we're not, if we're just trying to get a molecular description of it for use in Topaz, then we can just go through this process here. So what we're trying, just as a reminder, we've got our CIF file that is a coordinate-based system based on your unit cell axes, so fractional coordinates of a unit cell. So you are a fraction of the basis vector. So remember, your basis vectors are A, B, and C, your unit cell. Okay, they're called your basis vectors because there's a direction and a magnitude associated with them. And then each of our coordinates for our system is a fraction of A, fraction of B, and fraction of C. Now, in a single crystal experiment, we've got enough data for us to be able to do stuff with, whereas in a powder, we don't have very much data. So what we can do is we can, what we can do is we can, instead of refining each individual position, what we're able to do is say, well, this is what my molecule looks like. These are the A to B bonds, these are ABC angles and torsions. The molecule is not going to move away from that, really. There may be a torsional angle here or a twisting around a bond or something, but the bond lengths and angles are going to remain. So please just use this model and fit the model as best you can to the data that we've got. Okay, so this is what we're trying to do is we're trying to do what is termed a rigid body refinement. Okay, so the body is rigid, your molecule is rigid, whatever it might be. It's going to be in that shape, and all we're going to be doing is rotating it, translating it, and rotating it. Okay, I will switch off this stupid background. This thing's height. Choose. Video filter. Right. Hello. No. Okay. So none, please. Virtual backgrounds, none. There we go. So we've got this molecule here, for example, and we're going to just say this is what the molecule is going to be. In single crystal, I could just say, can you please refine where all the atom positions are? But this is the molecule that I'm going to be for the rigid body, I'm inputting this, and all you need to do is translate it, move it up and down, and twist it around so it fits the data as best it can. So within Dash, we've got this new setup, simulated annealing runs, and we can click on to this. And here, what we're wanting to do is, I don't think you even need an SDI file then. Well, I guess you don't need to do it. Last week we did. For some reason, we don't this time. So what I'm going to do is I'm just going to go and repeat what we did here. Where are we? We are. Copy that. Paste it into there. Okay, so what did we do last time? GliActive. Okay, and so by inputting that CIF file, you can see that it's split out. There are two molecules in the CIF file, independent. So there are two molecules output here. Does that make sense?
Mm-hmm.
Yeah. So you can see that there's a Z matrix that is being produced. And in that file structure, you will see that I produced on the 8th of May at 1:10 I've got these two files here. Okay. Now, if we open this up, as a reminder, we've got this setup. We've got our carbon, and then 3 columns. That's going to be our bond lengths, bond angles, and torsional angles. And this is the way that it's set up in Dash. It's not the way I like to look at it, and I much prefer the Topaz one, so we'll get onto that in a minute. But you can see there that there's distances between various atoms and so forth, and angles. So you don't need a coordinate system for your Z matrix. You don't need to know what your unit cell parameters are. You can create a Z matrix out of anything. Okay, so if you dream up a molecule, then you can create a Z matrix of this, okay, quite easily. Now, what we do is we input that Z matrix into this program here. So if we click on that Streamlit, it might tell you about updating programs or something like that. Yeah. And here, for example, it gives you options by which you can look at a system. Don't know why it's not allowing me to move up. That's annoying. But essentially, I can browse from a Z matrix here. Yep. Or drag and drop. So I can drag and drop in there. Okay, so that tells me that's going to convert that Z matrix into something that Topaz is going to be able to read. It's asking you what the B factor is. That's the ADP, is your thermal parameter for your system. Okay, so that's the powder diffraction torque for ADPs, or your isotropic temperature factor, and that for your hydrogen. So they're going to be slightly different from one another. You could define just one, and then multiply this one by 1.5, which is what single crystal refinements do. But we can change this if we want to change that. But essentially what we'll get from this is, if I download it, if I download this, then what we've got is, there's it, INP file. Okay, so we've got our INP file for that particular molecule. And so you can see here that you've got your positions in terms of your basis vectors, your ABC. So that'll be your X coordinate, Y coordinate, and Z coordinate. You've got your occupancy of whatever atom it might be. So if, for example, you're doing neutron, then you might need to change these H's to D's. You've got an occupancy saying it's 1, so that all the atoms are there 100% of the time. And then you've got this thermal parameter over here. underneath here then we've got a rigid body, which is what we've just converted. So in here, this is the format of it. What it's saying is the carbon 3 to the carbon, that's the reason. You have it in reference to one of these atoms. Okay, so for this molecule here, I might have this N group here as my atom to start with, this one here. And so I would say O1, and then carbon is next to O1, carbon is next to carbon is next to O1, and build up our model based on that first initial atom. So in this system, we've got a Z4, what is it, C4Z. We're saying that C3Z